Well, welcome everybody to Engineering 340 E4 542. At the moment, we're going to try to do this jointly online and in person. The majority of people that I asked seem to prefer to do it online only. And we'll see how things pan out with that, okay? If you have very strong feelings about it one way or the other, please do let me know, and we'll try to accommodate. Unfortunately, I think it's reasonably likely that we're going to wind up online only no matter what, relatively soon. So we did it that way last year and it worked out pretty well. So let's see if I can do this. Okay, so I want to talk about today I'm going to talk about a little bit about why we're talking about viruses as a theme of the course. And I should say that the methodologies that we're going to develop in this class are ones that could be used for a whole variety of topics. Viruses are one. And there's no reason that in class projects and things like that, that you have to stick to viruses. You're free to do what you like. We're going to talk a little bit generically about what networks are. I know one or two of people come from the Network Science Institute, so that should be familiar to people. We're going to talk a little bit about how the class works, just logistics. Start talking about dynamic network models, which is what the class is about. And then we are going to try to get in and actually build some models. And so then we'll need your laptops, ideally, to work on that. And so that's the theme of the class today. Clearly, this is a bit of an out-of-date slide. The world is telling us that viruses are important. We're going to start out by talking about epidemiological models. How viruses spread in populations. But we're going to find out that the way viruses spread within the body is actually pretty similar. And so all of the things we're going to learn about epidemiological modeling are going to transfer over to modeling what goes on inside of the body. And as I say, the methodologies that we're going to use are pretty generic. They're going to work for a whole variety of problems, not just viruses. We're going to be talking about normal times. There's always a huge crowd out in that hallway, and it's very loud in here. I guess that's less down with the virus. So we're going to talk first today about what we can do with network models. And there's quite a bit. Not everything in the world can be modeled as a network, but many things can. How we build dynamic network models, what their basic properties are, and then we're going to talk a little bit in this class, not too much, on the mathematics of it. If people are interested in the mathematics, we could go over it in more detail. But depending on the year, some people have more mathematical interest, some less. Again, we'll try to introduce all of the ideas that we use in the class as we come to them, but some of them will be presented relatively quickly. So if it's something that's unfamiliar to you, we could either give you background material to look at, or we could go over it in more detail. So don't feel shy about saying you don't know how to do something or you haven't seen something before, and ask for more information. We're going to start out by asking the basic epidemiological question, which is how do viruses spread in the population? And then we're going to move on to what are called in-host viral models, which is how does a virus spread within your tissues and how does your body eliminate it? We may talk a little bit about how viruses replicate within individual cells. So we're going from a population scale to an individual scale to what's going on inside a single cell. In my own research, we do a lot of cross-scale modeling. In this class, that's something one might be able to do in a project, but it's not something we're going to talk about too much in the individual lectures. We're also going to talk at least 1 or 2 classes about distribution and clearances of drugs in the body. A lot of modern pharmaceutical companies, in fact, almost all modern pharmaceuticals are modeled using what are called PBPK, physiologically based pharmacokinetic models, which model when you take a pill, how long does it take to get into your tissues? How long does it last in your tissues? Which tissues does it go to? These kinds of things. And then we could look a little bit, if we look at previous class projects, a lot of people looked at questions about effectiveness of different drug treatment strategies. There are a lot of other classical applications which we've covered in other years, how biological chemical synthesis pathways work, how cells signal and receive signals, how cells differentiate during the development or de-differentiate during cancer progression. People who worked on neural networks, not so much kind of AI neural networks, but more modeling individual neurons. And all of these can be modeled using the same mathematical structures. And that's what makes this kind of approach very powerful. In the class, we're going to use a single simple modeling package called Tellurium, which was written by Herbert Sauro at the University of Washington and Andy Samoji here at IU. Andy has moved on now. He now works for the military, but he was the co-developer of this package. It lets you get your hands dirty and write models very, very quickly and simply. And in fact, the whole description of the language is in 1 page, which I've handed out here, that Tellurium cheat sheet. We're typically going to be running the Tellurium and antibody models online in a public server called NanoHub. You can certainly download Tellurium and run it on your local laptop, and there's some convenience factors to that. The disadvantage of doing it that way is that depending on whether you're a Mac, a PC, a Linux box, whether you're using Jupyter Notebooks or something else, you'll see something different. And if we all use the NanoHub deployment, then everybody sees the same thing. So all of my teaching is going to use NanoHub. And so if you decide to run it locally, you're going to have to make your own mental adjustment of how to adjust that. We're going to do some fitting, plotting, and data analysis using basic standard Python packages, NumPy and SciPy. We will come to those when we need them. In general, we'll always use NumPy because Python without NumPy is sort of brain damage. Maybe at the end of the class, if Giuliano is willing to help us out, we could do a little bit of AI work, and he could show us a little bit of that. In terms of programming, this class does not make very high demands. You will need to learn a little bit of Python, but the most complicated things you'll be doing will be plotting, having to know matplotlib calls, which I never remember. I look them up when I need them. And also how to do array manipulation, because you have to select columns and these kind of things. But in general, we don't do anything very sophisticated. In your projects, you're welcome to do things as sophisticated as you like, but this is meant to be a gentle introduction, so we try to keep it that way. If it isn't a gentle introduction, please speak up. Again, the main things I would send you to right at the moment are the Python and Tellurium cheat sheets that I handed out here that I've sent around to people. There is an online textbook and tutorials for Tellurium at telluriumreadthedocs.io. I think it's really a great introduction. It covers a lot of the material that we're going to cover from the perspective of chemical reactions. And so all the methodologies will be the same, but you'll be seeing a different deployment of these same tools. And so I encourage you to take a look at that. And again, I often look at those examples when I'm looking for methods. If you've never done any Python programming at all, and you want to learn Python quickly, I really love the Rosalind online package. It's a free package here. It teaches Python programming in the context of bioinformatics, which is rather interesting. And it has starts with nothing and takes you through very sophisticated Python with graded exercises. And so it says, write a little program to do this, and then it gives you a data set, you plug it in, and it tells you whether you got the right answer. If people have not learned Python and they need to learn it, I give, and you complete the Rosalind exercises, you can get some extra credit. So if you send me the documentation that you've done them, you're welcome to some extra credit for that. Again, we're not going to use a lot of Python, but I really do like that package a lot, and it's free. So let's now talk a little bit about networks. Our lives are full of networks. They're very general organizational concept. They can be used to describe everything from, well, we'll see a bunch of examples in a minute, but many things in the world can be thought about as networks. And we're going to be focusing in the class on biological networks. We're going to be focusing on the subcategory of viral infection-related networks. But again, I want to emphasize that these are methods that you could use in a whole variety of contexts. It's not limited to biology or to viruses. And so let's look at some examples of networks. Boy, some of these are out of date. But a classic network, and one that's not irrelevant to viruses, is the one on the top right, which is a very old map of the United Airlines route structure, showing the main cities that United Airlines served and the flight routes that connected those cities. So they've labeled their hubs. You could probably date it by which hubs they've got. Cleveland isn't the hub for United Airlines anymore, so it tells you it's an old picture. And that is something that represents something that's not real, is partly in real space, because the cities really have locations. On the other hand, the connections between the cities aren't real physical objects, they're conceptual links between the cities. Another example of a network, if anybody has talked to Connie Berner or worked with Connie Berner here in informatics and SLIS, would be a citation network. So on the bottom left, somebody has looked at topics related to philosophy and looked at how those cluster, how a paper on one topic cites a paper on another topic, it cites a paper on another topic, to try to give an organization to the conceptual ideas of philosophy. Yet another network, and this is also pretty out of date, would be the physical connections of high-speed internet backbones in the US. So those could be fiber optic cables that are connecting internet relay centers. Again, these things go out of date almost before they're printed. So these slide is relatively old. But yet another one would be this picture here, which is typically not very informative. It's what's called a hair bowl, is what's called a protein interaction network. So in that case, somebody has developed, this is again fairly old, somebody had developed technologies to ask the questions, looking at the proteins in the cell, I believe those are bacterial proteins in that case, which proteins interact with which other proteins. So each one of those dots represents a protein, each line represents an interaction. And one of the things that you'll see rapidly when you look at these networks, a network that's embedded in real space, like the internet network or the airline network, makes some sense when you draw a picture of it. The citation network or the protein network don't really embed in two-dimensional space very well. And so you get what's called a hair bowl. You just get a lot of points with a fuzz of lines. You can't really see what's going on. In this class, we're going to be dealing with networks typically with relatively few nodes and connections. not hairballs, although occasionally people have tried to do hairball type projects. Here's some more networks that we could talk about. And one can think about how these networks are similar and how they're different, because there really are important overlaps and also important differences between them. For example, one question is, are the connections directed or not? Is there a directionality in time or in space to the connection? Or what do the nodes represent? In the bottom left, we have a human disease network, which is talking not about the names of diseases, but rather about the kinds of problems they cause. Does the disease affect the lungs? Does the disease affect metabolism? Does the disease affect muscle? And it's clustering diseases that way. On the top is an economic network. It's showing trade relations and exchange of economic materials. And on the bottom right, we have a phylogenetic tree. So this is an evolutionary network. Clearly, again, there are significant differences in terms of what we're talking about in each one of these. Here's some more networks. Here on the left, top left, is a classic epidemiological network. In this case, it was influenza transmission. A number of years ago, we had here at IU Alessandro Vespignani in informatics, who was one of the premier modelers of influenza epidemics. Several of his students wound up working for Bill Gates after they graduated, actually, directly. But here you have an imaginary network where you have a person who's infected, he infects some other people, they go on to infect other people, they create chains of infection. And of course, if we're going to be talking about COVID in detail, we'd have to do that kind of model. Again, in this class, we probably won't get there, although it would be a good class project. Bottom left is an RNA silencing network that's going on again inside a single cell. That's about that model of network regulation inside individual cells. At the top is an ancient and famous picture of neurons in a brain, stained neurons in the brain, so a physical network of neurons. And then below it is the classic structure of a simple artificial neural network. And one could ask the question, how does that physical network of neurons relate to that computational neural network below? So those are all networks. When we come to biological networks, classically, biological networks have four main types when you go inside the body. Those types are known as chemical reaction or metabolic networks. Those typically inside individual cells are the pathways that build and break down the chemical components of the cell. There are also signaling networks that transfer information outside cells to inside cells and within the cell. They relay information typically from the cell membrane into the cytoplasm and then into the cell nucleus. And in pre-COVID days, most of this class would have been spent addressing chemical networks and signaling networks. That's certainly an area of interest to me and something I worked on quite a bit in my own work. The third one down are what are called regulatory networks, and particularly gene regulatory networks. Cells turn genes on and off, in particular during development, but also in response to external chemical signals and contact signals in the history of the cell. During development, you start with a single fertilized egg. You have to build an organism with lots and lots of cell types. So genes have to be turned on and off. And so these gene regulatory networks are something that we run into. When you're talking about things like viral replication, you have to take, the virus has its genome, the genes of the virus have to be transcribed and then translated. And so that would involve both chemical reaction networks and gene regulatory networks. And then I've already mentioned this case of a whole body physical pharmacokinetic models that are used to describe the transport within the body, primarily the flow of blood within the body, and the various organ systems that are involved. When we think about other kinds of networks, there are what are called population dynamics models. That's what we're going to run into when we do simple epidemiology. In this case, I'm showing it in the context of your gut. This is a picture of a colonic crypt. And at the top, you see at the bottom of each colonic crypt, your gut, if you've ever eaten tripe, you know what it looks like. At the bottom of each crypt, you have a number of stem cells. Those stem cells reproduce. They push cells away from themselves up the walls of the crypt. Those cells in turn divide, differentiate to form semi-differentiated cells. And then at the top of the crypt, you have non-reproducing cells, which are secretory cells, which secrete the mucus and other things that go into the gut. In the case of a colonic crypt, the cells have a lifetime of about 2 weeks. So it takes about 2 weeks for the cells to go from the bottom to the top. And models, for example, of leukemias are built like that. And we'll find that also the models that we build of epidemiology are going to look a lot like that. And then at the bottom, we have a toxicological network where we have some rather abstract concepts about how damage occurs. And some models of cancer also are like that, where you have a sequence of events where you, for example, expose a cell to a toxin. Actually, to be correct, it should be called a toxicant. That toxicant causes some change in the cell. That change leads to other changes, and you have an accumulation of damage that then leads to a macroscopic adverse outcome. And so all of these are networks of one kind or another. As you can see, a network like the one on the top right is a very simple one compared to that hairball of the United Airlines flight map or the protein interaction network. And so one of the things that we're going to have to do repeatedly in this class is decide what level of detail we need to use to describe a particular phenomenon, whether it's a biological or other phenomenon. Okay, so I hope there's any questions about that so far. Okay, so to try to understand networks and their context and meaning, we're going to be pulling a little bit of information from a lot of fields. And so you don't need a lot of specialized knowledge about any single area, but you are going to need to know a little bit about a lot of things. You're going to need to know some biology, a little bit of biochemistry, some bioengineering, a little bit of biophysics and chemistry, a little bit of computer science and data science, a little bit of the mathematics, dynamical systems and general mathematics and statistics. And in general, unless people have taken this class before or taken a comparable class, I don't expect people to come in having all of that. And over the years, I've had people from the Department of Mathematics take the class, from bioengineering take the class, from psychology and brain science, chemistry. And so each person comes in with their own background. And one of the things that I depend on you to do a little bit is to try to fill in your gaps. And it may take time to figure out what they are, but in general, you don't need to be an expert. I say typically a Wikipedia level in knowledge of these topics is probably enough for most things. And when it's not, we'll try to go in. And if you need help in any area, please come to us. Well, actually, to me, this year I'm the only one. We don't have a TA, so it's me. Although Josh Aponte Serrano and Giuliano have agreed to help out, who are very expert in this. So 2 of the graduate students in my group have agreed to help out. So they're available to answer questions too. And 2 points. First, if something is confusing to you, it's probably confusing to the other people in the class. So please make sure you ask about it. And secondly, if it's confusing to you, there may actually also be somebody in the class who knows something about it, depending on background. I mean, in a year when we've had an experimental biologist, they can tell us about the details of some of the biology that other people might not know, or if you have somebody computing. So also feel free to speak up. If you know a lot about something, speak up. If I say something that's wrong, don't be shy about telling me that I'm wrong. I'm not a specialist in most of these areas either, but so I'm quite capable of making my own mistakes. One of the things that we'd like to talk about a little bit is why this is a class in engineering. And this class grew out of a class that I taught many years ago in physics. We're focusing on mathematical biology. And there's a lot of overlap between biophysics and bioengineering, computational biology, mathematical biology. But they're not quite the same. And so if you want to try to do a rough idea, you could say that science is about deriving and discovering models or governing principles that predict the behavior of natural systems under particular conditions. And so science is really about prediction. Now, engineering requires prediction because if you're going to design something, you have to be able to predict what it's going to do. If you can't predict what it's going to do, you don't get anything out of it. On the other hand, engineering really goes a step further than scientific understanding in the sense you're trying to take the scientific understanding and use it either to build new things or to control things that exist. And so engineering, to a certain extent, is about design and control, whereas science is more about understanding and prediction. And again, they're not totally unrelated to each other. They're very closely connected. But this really is going to be more about control. When we talk about science and engineering in biomedicine, we first want to be able to understand what the processes are that result in the phenomena that we see. And typically this is going to be something crossing scales. So we'll build a model, say, of what's happening inside a single cell and ask the question, how, if we put a lot of those cells together, do they make a tumor or not? Does it make a cancer or not? We put a lot of individuals together, one of them is infected, what happens at the population level to the infection? We put a single virus particle into your lungs, it infects a single cell in your lungs, what happens after that? And so those are the kinds of questions we're going to ask. Ultimately, we want to be able to predict new phenomena. We want to be able to identify what the key mechanisms are. Especially in biology, we don't have any first principles models. We're always building phenomenological simplified models. And so we want to be able to understand what the things are that we need to use and what we can leave out. And a lot of the art of modeling is about deciding what you omit, what you leave out. Clearly, if things are going to be useful, you'd like to be able to bring them back to the real world. And so you'd like to either be able to suggest new experiments or help interpret existing experiments. There's some good practical reasons for that. Experiments are expensive. You can't do that many of them, and they don't give you complete control or the complete ability to watch what's happening. Especially in medicine, your ability to do experiments on human beings is very limited, and your ability to monitor human beings is very limited. One of the things we find if you look at COVID is that even the most basic thing, you ask the question, when you're infected with COVID, what is the amount of virus in your lungs as a function of time? And you will be maybe surprised, maybe not, to find out that the number of individuals in which that's been measured is very few, and the detail with which it's been measured is not good. The amount of virus in your lungs when you're infected with influenza or COVID can change by a factor of 1,000 in a day. Typically, if you're lucky, they measure the viral load in your lungs once a day. It's inconceivable in any other field that you'd make a measurement every 1,000-fold change in the thing you're trying to measure. And so the people who do COVID modeling actually face the fact that there is not very good data on COVID. So that's another kind of thing that you might think about. Ideally, you would like to be able, if you're dealing with a disease, you'd like to be able to improve therapies for the disease. And you'd like to be able to guide policy. And you'll hear a lot of argument in the news. The model in England or Australia, the model for infection. In Australia, there was just a discussion in the news in the past couple of days where there was a model, epidemiological model, that said that if Australia unlocks with 80% of the population vaccinated, they could have 100,000 deaths over the next year. The government said they disagreed with that. They're going to unlock no matter what. So there's a difficult connection between political decisions and scientific decisions and medical decisions, especially when we deal with epidemiology. I'm a co-lead of a working group with NIH that discusses some of these issues, and we've had some nice speakers. Those lectures are all available online. I can send you the links if you're interested. And we've had some people talk about the policy implications of modeling. Generally, when you build computer models, especially the simple ones that we're going to do, they don't wind up becoming political discussion points. But in fact, when you do epidemiology, it's politicized. So that's not something we'll really address in this class, but it's something people need to be aware of. So again, this course is in engineering because engineering requires us to predict things. And if we want to be able to design things, we have to have decent prediction. And so a classic example, of course, with epidemiology would be the design of social distancing rules. Why are we wearing masks? Why are there in-person classes? What's the effect of what's the effect of social distancing or of vaccination when the vaccine isn't perfect? There are a lot of issues. What's the effect of switching from the Alpha variant of COVID to the Delta variant of COVID? How many people are going to be infected under each one of these situations? Rather important. Or if we have an antiviral medication, how do we use it effectively? Okay, so that's the background to get started. Let's talk a little bit about logistics. You've got me. You've gotten emails from me. If you haven't gotten an email from me, that means you didn't register for the class, because everybody who's been registered. So if you haven't registered for the class that you're interested, and you haven't gotten an email from me, please send me an email. I'll put you on the mailing list. I use my Gmail account for everything. The IU mail has a tendency to put important messages in spam. And so I prefer to use the Gmail service, which is more reliable. Joshua Ponte Serrano, who has TA'd this course many times, is not TAing this semester because he's leaving for the National Institutes of Health, NIAID, the Allergy and Infectious Diseases Agency, in November. So he can't TA this semester. But he's, as long as he's around, he's happy to help. He's an expert. And Giuliano Ferrari Gianlupi has done a lot of this work in the past and is also willing to help out. This semester, unlike the spring, we have 16 weeks of class. Last semester, we only had 14. That helps out. The plan is that we're going to at least start out with in-person and Zoom. Again, I think it's quite possible we'll wind up Zoom only pretty soon. If people are comfortable Zoom only, we can do that. It's easier probably for some of us that way, but let's see how it goes for a week or two and then we'll figure it out. And if people want to meet with me, I don't have regular office hours, but I try to be available. The other thing that I will say is that I get literally hundreds of email messages every day. So if I don't respond within 48 hours to an email, call me. My emails always have my cell phone number on them. Don't be shy about calling me. If I'm asleep, I won't answer the phone. So, but I'll at least, it does put you up to the top of the stack so that I know that you're looking for me. So if you don't get a response right away to an email, don't assume that I'm ignoring you intentionally, but it is possible that I didn't see it because it's lost in a stack of emails, especially at the beginning and end of semesters when things get hectic. Historically, the main textbook for this class is Herbert Sauerow's Systems Biology, Introduction to Pathway Modeling. That is focused primarily on molecular reaction signaling networks, but it's built around the tellurium modeling environment, and it's really a great book. It's not very expensive. I really encourage people to buy it. There's also a PDF. You can buy the PDF for not very much money as well. There's, if you want to learn a little bit more about the mathematics, some of which we'll skip, I like very much Nicholas Britton's Essential Mathematical Biology. It's a paperback. You can get it secondhand, not expensive. It's probably the best, most compact introduction to simple mathematics that we need. There's a textbook called by Eberhard White, First Course of Systems Biology. That focuses more on metabolic networks. It's a little more sophisticated than what we'll do in this class. But if you're interested in those things, it's not a bad thing to look at. Herbert has some other textbooks as well that go into more detail. Last year, we recommended a book by Brower and Chavez and Feng on mathematical models in epidemiology. It goes into quite a bit more detail than we'll do here. If you're interested in epidemiology, it's not a bad thing to have. I think actually, because it's a Springer book, you may be able to get it for free through the university. Yeah, then there's some additional references. There are a couple of papers that I strongly encourage you to read. In particular, there's this nice recent paper called Your 10 Simple Rules for Your First Mathematical Biology Model that was written by some students in Canada. That's a very good introduction to some of the ideas that we're going to have. If people find papers that are useful that we should include in our stack, I appreciate that very much. Course logistics, people usually want to know about grades. They're going to be weekly problem sets, which will be 50%. The main work of the class will be a mini project, which will be 30%. They'll do an oral presentation of that. That'll be 10%. Class participation is 10%. I'm not exactly sure what that means. Basically a way for me to give you a few extra points usually. If you don't show up, then that's maybe that's an issue. Again, if you need to do some background work on Python or Rosetta or Python or programming in general, I do complete the Rosalind course or Code Academy course. I'll give you some extra points for that. There's no final exam. There's no midterm anymore. But I do ask you to try to get the homework assignments in on time. Occasionally we'll slip because I don't always know exactly how much we'll cover in a week. So the assignments are written in advance. And sometimes life happens, but please try to get them in on time. It is, while the material isn't difficult, there is a fair amount of work to just keep up with the things that are being done. And so if you get behind, it's hard to catch up. I will try to upload things to Canvas. Maintaining 2 copies of everything is always a bit of a pain. In general, the primary source for materials will be this Google Drive. Does everybody have the link to that Google Drive? Okay, if you don't, let me know. Let me see if I can put that in the chat for the people who are joining us remotely. Okay. Okay, I strongly recommend, as I said, a Wikipedia level understanding of the basics. They're good Wikipedia articles on network theory, network science, dynamical network analysis, COVID, the immune system. We created a folder called papers. It's actually not called papers to review. It's called papers. I have to look at the name. The title is slightly different. Papers review. It's in the student materials directory on Google Drive, so you'll find it. And as I mentioned, there's this recent article on 10 simple rules for tackling mathematical models, which I think is pretty good. You may find it helpful. A lot of the classwork will be related to finding material and bringing it in and talking about it. This is a participatory class. And so your help in coming up with good things to talk about would be appreciated. I'd like to talk a little bit about the term project. I know the end of the class seems a long way away. When you're actually doing projects, it's not as far away as it seems, especially when it's new material. One of the tensions in this class is that starting at the beginning of the class, a lot of people don't know what the class is about. And that makes it difficult to come up with a term project. On the other hand, if we wait till the middle of the semester for you to pick a project, then you're not going to have time to do it, because these projects are things that can't be rushed. And I prefer, if at all possible, to not have people get a lot of incompletes in their classes. Those also happen, but you probably prefer it, but I probably prefer not having it. And so I'm going to try to push you to try to come up with a project idea as early as possible in the class. And if that's impossible and you change your mind in the middle of the semester, we can handle it, but I still want to push people to do that. When this class started, people by default seemed to want to work as pairs or triples in small teams. In the past 2 years, for reasons that I don't quite understand, people have preferred to work on their own. I encourage people to work in teams, but I will not absolutely require you to work in a team, especially if you have a project. If you really want to do a project nobody else is interested, you're sort of stuck on your own. But very often, and last semester was an example of this, we had a number of people who looked at vaccination strategies for COVID. And we wound up having 3 separate papers on vaccination strategies for COVID, all of which were very good, by the way. But it might have been easier for people to have worked together and come up with one super paper on vaccination strategies. So I do encourage you to try to find a classmate or two to work with. In particular, if people have complementary backgrounds, somebody's very good at computing and somebody else is very good at biology. Somebody knows a lot of math, somebody doesn't know much math. Those kind of teams can be really valuable working together. The project will probably, and again, this has evolved a little bit over the years, but typically the project, at least the sort of the minimal project, is to find a paper or two that address a topic that you're interested in, reproduce it using the methodologies that we teach in the class, and then you're going to deploy it as a learning tool on the NanoHub site. And I will show you in a minute some examples of previous class projects so you can get an idea of what they look like. The hardest thing I find in the NanoHub sites is that it's relatively easy to write a term paper about your project. But, and this is also true when people try to write textbooks. The hardest part of a textbook is not the text, it's the exercises. And NanoHub is designed to be an educational resource where people come in, they read your paper, the figures in your paper are live simulations. And they run those simulations and they learn something. And so the hardest part usually of these term projects, is coming up with exercises that people learn from. And one of the best ways to demonstrate that you understand it is to be able to teach people. And so I'd like to suggest that. And let me see if I can actually get one of these to run for you. And if it doesn't work, we'll just have to live with it. I'm pasting the link into the chat. Let's see if NanoHub is going to play nice today. All right. Okay. So this was a class project from last semester by one of the undergraduates here in informatics, in ISE, Antonio Nico. And he was interested in tuberculosis. So not a viral infection, but a bacterial infection. One of the issues with tuberculosis is that it's actually very hard to get rid of. There is a vaccine for tuberculosis, but it's not very effective. It's not used in the United States. It is used in Europe. So whereas the COVID vaccines are at least pretty effective, the tuberculosis vaccine is marginally effective. Antibiotic treatment clears primary symptoms pretty effectively, but actually to clear tuberculosis completely, you typically have to, oh, here I am, sorry. Excuse me. Here I am talking and you don't see it. I did, hold on a second. Sorry about that. Sorry, guys. Our online people are seeing the right thing and the people in the room aren't. Let me see if I get this to work. Sorry, let me reshare. So now you're seeing it and the online people aren't. So now. Back at home, I have my multiple screens set up so I know exactly how the layout's work. Here I have to do this. Okay, so let me try that again. Online people can see it okay? Yeah.
Yes.
Welcome. Great. Okay, so here's Antonio's project. So what did he do? He has a discussion about tuberculosis and why tuberculosis is an issue. Some of the key problems that you have to address in tuberculosis, namely that most people who start the antibiotic stop taking the antibiotic before they clear the disease. In fact, tuberculosis in general is something once you're infected with it, you'd almost never get rid of it completely. People stop taking the antibiotic because it makes them fairly sick and they feel better quite quickly, and then they don't keep taking it. The other problem with stopping to take the antibiotic, besides the fact that you're still carrying tuberculosis, and that means that if you're ever immune compromised, you can break out and die from it, is that it induces drug-resistant tuberculosis. And in a lot of the world, especially in Southeast Asia and South Asia, the tuberculosis strains are mostly resistant to the primary antibiotics. As we know, also in the West, the overuse of antibiotics has led to a lot of antibiotic-resistant bacteria. And so this was a model trying to include clearance of the disease due to treatment and the development of resistant strains. Okay, he looks at what happens in the population if you start out with people who are uninfected. This is an epidemiological model. Blue is uninfected. Red is infected and infectious. Yellow is a latent carrier that's not infected and not infectious, but still carries the disease. And green is somebody who's cleared the disease. And then he looks into the various aspects of the disease, describes what you see, and you'll get a lot of homework problems that say things like describe what you see. But I realize that open-ended questions like that can be confusing. So please feel free to ask for clarification about what it means. But here he's done a very good job explaining what that time series means. Let's look. Notice how long it takes for a population to start seeing rising cases, because time-scale tuberculosis is very slow. Okay, and then he has a variety of explorations of different aspects of the disease. And now you have what is nice to do in the Jupyter notebooks, which is those are pictures just like you'd have in any scientific article or term paper. But what you can do in Jupyter notebooks is you can put into your article simulations. And so this is a simulation where it's not doing that much. You can ask the question, if people encounter each other, the more people you encounter, if you're socially distanced, the disease spreads less. If you increase the number of interactions that people have, let's see if it works. Boy, so let's start out with this. This is actually a number of infectious interactions per day. So this is the baseline where the infected person has 1/2 every 2 days they interact with somebody who could be infected by them. And you see the blue curve, half the population gets infected after 20 years. It's a slow process. Now I say, what if they interact with 1 person a day instead of 1/2 a person a day? Now 90% of the population gets infected, and it takes 5 years to happen. If I go up, they interact with 2 people a day, 95% of the population gets infected, it takes even less time to happen. And so you could build a simulation in this case where you explore how the parameters in the model affect what's going on. And then he's explained what you see and what you need to change to learn about what's going on. Here he's looked at resistance. And you could say, okay, what happens if you have drug resistance? In this case, 65% drug resistance. We increase the drug resistance. Actually, the number uninfected goes, sorry, this is the other way around. Percentage resistant to reinfection is actually the opposite of what I said. This is that if you recover, this is how long your antibodies last. So higher number means you're more unlikely to get infected a second time. If you have a situation where you lose resistance rapidly, then the percentage of people who get infected goes way up. And so again, he has a whole series of explorations. Here he's got a model of vaccination. He explores a bunch of different vaccination strategies. And you can see how that works. Okay, he gradually builds more complex models as he goes through it. So I'm not expecting that everybody's term paper is this elaborate, but this is an example of what you could do with this kind of display. Okay. I don't know, there was one other one. Yes, you did. Again, it's not, it's great when people do that much. It's not the standard of the class. last year we had a bunch of students who were really very committed to this class, who wanted to go to MD, PhD programs. And they wanted these projects to be things they could show off in their med school applications. We've also had students who use their projects from here to get jobs at Facebook, Google, those places too. Again, that's not required. I mean, the expectations here are reasonable, but it's sort of open-ended in the sense that some people, well, you have a limited amount you want to learn and you learn it, that's fine. Some people find it something they really want to do. It's been really a pleasure to me when it's not that I don't want to devalue any student, because I value all students who participate. I'm grateful to you for being here. But I also want to say that occasionally we get students who have great ideas. And it's fun to see people explore and come up with neat ideas. Not required, but is possible. Just to show that there isn't, it's not only infectious diseases. Let's see if I can get this one to work. One thing that's another confusion with using the Zoom here is that even though that screen is on my left, logically the computer thinks it's on the right. So the mouse moves backwards. The mouse moves backwards on this, so it's like a mirror. So this was a project that was done by another student in the class. He was a master's student here. Let me make sure that it's being shared. Yeah, okay, that's fine. So this is a model of synchronization of neurons. So in this case, neurons fire action potentials. You can model how those action potentials work. At some level, you can think of a thought as that these neurons fire periodic, or actually not completely periodic, but quasi-periodic spikes, grains of spikes. In some ways, having a thought consists of the localization of neural activity and the synchronization of neurons. And so this was a simulation that one of the students did, trying to understand how how neurons synchronize. He started out with a model of the ion channels in individual neurons, a simple Buffett model, but nevertheless a model of ion channels. And then he asked the question, if you change the interactions between the neurons, do they synchronize or not? And how does the frequency of the oscillation change as you change the parameters of the simulation? And I have to say, for the other one, I know what the parameters do and mean. This one I didn't review as much before today. I really just wanted to show. He did some nice 3D plotting where you could use interested dynamical systems, so you could actually explore the what's called the phase portrait of the neural network. So I just wanted to show you a couple of examples of these. You will have the lists available. Here's one where you could see these. Here are two oscillators that are desynchronized. And now if we couple them, let's see if it works. Did it do anything? Maybe not. Can't promise everything works. See if this one works. Yeah, this one works. So you have three oscillators that are independent of each other. You change their coupling. And in principle, if I do it right, I should be able to see them lock onto each other. I'd have to read the article to find out how it works. Okay, So that's just an example of how one can look at different kinds of problems. And depending on your background and interest, you're free to explore. Okay. So we have a directory in the student materials folder which lists possible papers to look at. Please suggest more. The idea is not that those are meant to be exclusive. It was just to get you started. Don't feel that because we cover epidemiology at the beginning of the class that you have to model epidemiology for a project. And we've tried to create a Google Doc which has a little bit, the list of papers. They're categorized, but I realize that there are a whole bunch of things that we should have on that list that aren't there. So a little bit of Googling is really a good idea for this. So the timeline for the term project is for next week already. I would like you to go online, find one paper that has something to do with networks, this kind of ODE modeling that we're going to talk about, and write a paragraph or two describing it. And I've given you some guidance for that. In 2 weeks or a little bit more, maybe, I'd like you to come up with a list of papers that you think are interesting. And then by the middle of September, if possible, again, this might slip by 1 week, depending on how we do. I'd like people to have a sort of a one-page preliminary proposal. And I'd like everybody to have a couple of slides, like 1 or 2 slides that they can present to the whole class. And that way, if you find out that there are 3 people who are interested in the same thing, you can form a team. And then on the just before Thanksgiving, I would like people to have a sort of a draft of the paper and the application. And this year we're going to try things a little bit differently. In previous years, I've asked people to write in class comments for their classmates. But in fact, that's not a very effective thing to do. So what I'd like to have people do instead is dig into one. I'll assign each person to review and run somebody else's project and write a little bit more detailed critique. And then you'll have 2 to 3 weeks to revise it and upload it. In my experience, these NanoHub apps, the first version is always okay, but they really improve by being rewritten. So it's a discipline. And so I'm going to ask people to do that. So let me come down to talking about, so that's the logistics of the class. Are there any questions about that? And if you're online, you're free to either break in and ask the question out loud or use the chat. I have the chat open. Okay. Please.
Yeah, so for like the due dates, is it due before class for that day, or is it like
If you want to send it in at midnight of the day of class, it's all right. I'm not going to look at it after 8 p.m. today. So
like for the assignment next week, that's the day first. You want it done like or 5 Tuesday or like 5 midnight on Tuesday.
I think the official, I mean, I'm not sure how it helps you very much because you're going to be in class, but if you need an extra few hours after class, you're welcome to clean up your exercises. I mean, we will sometimes go over homeworks in class, but typically I do that the week afterwards. And the reason for that is until I see what you've done, I don't know where people are having problems. And so if I give out an assignment and I see, gee, a lot of people are having trouble with some concept, then the following week, I'll add some slides and review the problems. But that doesn't usually happen until a week later because I don't know what the issues are. The class would be really boring if all we did was go over homework assignments. And so on the other hand, if there's a key idea that people are having an issue with, I want to make sure that you get it. You'll see in the homeworks that I'll say there'll be an amount of time, an approximate amount of time that I expect people to work. And sometimes I misestimate those things, although most of these assignments have been assignments that I've done before, so I have some idea about that. So the other thing is, because they're fairly open-ended, in principle, a lot of these assignments you can spend a lot of time on. And I'm not going to tell you not to spend a lot of time on it, but I don't expect a lot of time. So I do expect people to think. And I do expect people to take the assignment seriously. On the other hand, if you find yourself, if I say this assignment should take 4 hours and you spent 18 hours on it, then there are two possibilities. Either you're interpreting the assignment to be more demanding than I intended it to be, or there's an issue about understanding what the question is. And in both of those cases, I would encourage you to ask before you spend an enormous amount of time, unless you want to spend the time. In general, I like it when everybody has 10 out of 10 on their homeworks, but I don't expect people to have perfect assignments. I do expect people to make an effort on the assignments and to hand them in and to try to think about it. But the assignments are really more as learning exercises. They're not designed as torture apparatus. Sometimes it may feel that way, but they're not intended to be torture. Okay, so that's a very reasonable question. And there are going to be times when you'll have multiple assignments running simultaneously. And so when that happens, I will try to reduce the intensity of the assignments to balance that. But at any project-based course, there are going to be weeks when there's a lot of work and there are going to be weeks when there's less work. And one of the reasons I try to front load the projects a little bit is so that if you have other project-based courses, the last 3 weeks of the semester, you're not frantically trying to do 80 hours a week on all 3 projects. And because the modeling here is relatively simple and it requires a fair amount of background work, you can, once you've picked a project, you can get started on it pretty quickly. So let me talk a little bit. Does that help with that? Was that any? Okay. Okay. So I want to talk a little bit about some rather abstract things, which I hope will become more meaningful as we work with them. So we're going to use words like a model and simulation a lot in this class. And one of the things that I found is that those terms are a little bit of a minefield for people. And they can be a source of confusion. So what is a model, or what does a model do? Well, a model we could think of as a black box, although in your case, you're going to know what's inside of it because you're going to write it, that predicts something that you can measure. And ideally, that should be something you can measure in an experiment, because if you can't measure it, the output isn't meaningful. And it's going to depend on something that are going to be what we call a set of parameters, input parameters, that in principle, again, are things you could vary, or at least that naturally vary in the world, or that you could measure. And the other thing that a model has is what's called a model structure. And so, for example, if I look at in some years, we've actually spent rather a lot of time on this, and I do an exercise in class on it. The first exercise of the homework is going to be about this. So if I look, for example, at that United Airlines route map, that United Airlines route map tells me which cities have airplanes connecting. That is a model structure. It's an architecture. But it doesn't tell me, for example, how fast the airplanes fly, how many airplanes there are at any given time. Those are things that have to be added on to that basic structure, which would be parameters. And so there is this concept of a model architecture, a model structure. And associated parameters, and those together predict outputs. So, for example, in the example I showed of Nico's tuberculosis model, he had a model of infection. An infected person can infect other people, they can recover, they can die, they can become a latent carrier of the disease, and so on. That would be a model structure. The probability that they die every unit of time is a model parameter. The number of people who are initially infected in the population would be a model parameter. The number of interactions those people have with each other to infect each other would be a model parameter. And the prediction would then be how many infected people are there at each time step, how many people die at each time step, how many people have resistant tuberculosis at each time step. So those would be the output. So one of the key things about modeling that I mentioned before is that models are abstractions. They're phenomenological. They're not first principles. And that means that when we build models, we're leaving out an enormous amount of the complexity of the real world. And ideally, what we want is for models to be as simple as possible to make the predictions we care about. Now, we don't always know when we start out what that of selections is going to be. And that article that I recommended you to read in the student materials, it talks a little bit about how to do that. Selecting what to leave out in a model is very important. And it does require some experience and practice to be able to develop that. It's not something, there's no simple set of rules for that. There actually are some mathematical criteria that people like to use in some fields for defining what is a model that's sufficiently complicated or too complicated. They're based on basically Bayesian statistics. And one of the problems that we're going to face in biology in particular is that if there's a mechanism that you leave out, the model can't predict what happens when you perturb that mechanism. And so when you build very, very simple models, and we'll run into some of those, in particular, we talk about Alan Perlson's work, which is very beautiful modeling work of viruses. Those models are very predictive of the situations that he's experienced. They're trained on the data sets he has, but they can, and they're very good at interpolating between data sets. But they're not always as good at extrapolating, because when you go outside of the training space, you run into situations which may require mechanisms that weren't included. The kinds of models that my group and I build tend to be more complicated ones, where if we know a mechanism is there, we put it in, even if it's not that important in a particular situation, because we don't know the situations it's going to be used in. And so there's a little bit of again, that seems probably pretty abstract now, but I think as you go through the course, that may become more meaningful. So let's talk back to networks. Let's just say what a network is again. So a network is a model of a system, typically a graphical model. And networks have two fundamental elements to them. Nodes, which we're going to identify usually with circles or some other geometrical shape. And those nodes are connected with links, which are lines. And that is literally a topological network. It defines some structure, not necessarily structure you can embed in two-dimensional or three-dimensional space. And most of what this class is about is going to be making different choices about what those nodes and those links are, what they represent. Now, those links can be various kinds of things. They could represent somebody coughing on somebody else, which is a directed, it's an arrow, a directed node. They could simply be that two things are physically in contact with each other, which is undirected. They're different architectures. They're trees. We saw evolutionary trees, for example, as examples of networks. They could be hierarchical, they could be random. And there's a lot of mathematics and statistics that talks just about the structure of networks, just about the architecture of networks, their topology. Nodes and links have to have rules about how they interact. In general, a node could be connected to an arbitrary number of links, but typically links connect two nodes in simple cases. But when you're talking about chemical reactions, complex chemical reactions, you can have molecular species A, B, and C coming together and producing two species of output, D and E. Or molecular species A, B, and C producing one, D. And so links themselves don't have to be just a single straight line. There could be various complex arrows that show up. And don't worry, we'll deal with those gradually and build up to them. When we're doing science, typically we don't know what the network is to begin with. So a lot of biology in particular is doing what's called network inference. Genomics very often is trying to infer what the components are and how they connect. Proteomics is about understanding connection. A lot of biology is reverse engineering. We see a set of high-level interactions and behaviors, so we want to infer what network generated. In engineering, in classical human-based engineering, we design things, and that means we write down the network first, and then we ask what it does. And that's quite different. Engineered systems typically are in some ways easier to understand than natural ones. And we'll do a little bit of both in this class. Networks could be represented, as we've seen, could represent all sorts of different concepts. Their relationships could be very abstract, like citations between articles, journal articles, or they could be real entities like chemicals. They might have a spatial relationship like the cities in our United Airlines map, or they might not have any intrinsic spatial relationship like statement that two chemicals interact with each other. Of course, for two molecules to interact with each other, they have to come into contact. But the conceptual statement that two molecules can react is not a spatial one. So one of the things that's important to think about networks is that the network as a concept is not a spatial concept, it's a topological concept. It's possible to layer on spatial elements to networks, but it's not intrinsic to them, it's an add-on. And that comes up a lot. For example, I was talking yesterday with somebody who's a faculty member in England, University of Exeter, who works for a company called SimSip. And SimSip is a company, actually the CEO of SimSip gave a talk here a year or two ago to the undergraduate biomedical engineering students. They developed these pharmacokinetic models for drug companies. And what they want to know is if you swallow a pill, how much of the pill gets into your blood, which tissues does it go to, and how long is it there? Well, when you swallow a pill, first presumably you don't chew it if it's a swallowed pill. It goes into your esophagus, and then it goes into your stomach, and then it goes into your gut. Each one of those regions has a different temperature, a different pH. It has different mechanical properties. It has different ability to absorb chemicals. And so the simplest thing would just be to model pill goes into person, chemical goes into body, chemical gets distributed, broken down in the liver, excreted by the kidneys, and so on. But they were building more and more complex models where the entire gut, from the mouth to the anus, was drawn in little squares. Each one of those squares represented a part of your GI tract. And for each square, they knew what the temperature was, what the pH was, what its ability to absorb things was, whether it was mechanically distributing the pills or not. And so they were then able to build very accurate models about, if I change the mechanical properties of this pill, how does that change how you absorb the chemical? And so they were able to design what are called formulations that were more effective for drugs. So that's an example of taking a network. Yes.
I want to ask, like, what do you, I mean, like, I've heard the term topological before, but I don't really know what it means.
Okay. So a topological network, so topological means that what matters is what points exist and what they are connected to. But the length of the lines, the angles of the lines, and the positions of the points in space do not matter. So for example, if I take a balloon that's a sphere, that's topologically a sphere. If I squish it so it's flattened oval, it's still topologically a sphere, even though the shape has changed. If I poke a hole in the balloon so it looks like a donut, then the topology of the balloon is different. And so topology is about the structure, the connectivity of the structure of something, but not the specific positions of each component. Does that help?
Yes, thank you.
Good point. Okay. So as I mentioned, you could build models which try to overlay spatial structure onto a network architecture, but it does require some extra work. In general, we're not going to talk very much about spatial modeling in this class. In the spring, I teach a course which is focused on spatial modeling. Another thing that you can do sometimes is that typically the individual nodes in a model like this don't have any internal structure, they're just points. But one could build hierarchical models where each node itself is a network. So you could have networks of networks of networks. So for example, suppose that I had a network conceptually that were the cars driving in the city, and each one of those cars had a network that was controlling the car behavior. You have a bunch of Teslas that are self-driving, and there's a neural network controlling each car. So that would be a hierarchical network. By default, in most of the fields that we're going to work with, the nodes are the things that are being represented. Cities, airplanes, chemical species, and so on. And a node will have a state associated with it. That could be just its name, Bloomington, Indianapolis, Boston, or it could be a value. It could be the number of people living in Bloomington, the number of people living in Indianapolis. Or it could be something more complicated, like the number of men and women in the town between the ages of 13 and 15, or the fraction of people who are vaccinated. So the states could be potentially quite complex on each node. But typically, we're going to assume that we don't have internal structure in those nodes. The state can be complex. And if we were doing chemistry, that would be called a stirred reactor model. In fact, there's no internal state. But the node always has an associated state. A link, that is a line between nodes, defines some kind of relationship between those two nodes. That relationship could be something static, like I'm leaning against the wall, that's the relationship. Or in our world, we're going to be focusing on dynamics. We want to know how things change. And so in that case, the links represent interactions. And that means that an interaction is something that changes some aspect of node state. Typically, we describe interactions with arrows. And I have to warn you that there are a lot of different arrows that are used, and they're not always used consistently. And you have to be prepared to mentally transform one kind of arrow to another as you go from paper to paper or subject to subject. But typically, arrows have a concept of a source and a target, a single-ended arrow like this, or a bidirectional arrow means that there's an interaction going both ways. So if I can cough on somebody and infect them, then that would be an arrow coming from me to them. And the interaction would be by changing of their infection state. A bidirectional arrow could be by leading on somebody, I'm pushing on them and they're pushing on me. And there are a whole variety of arrows in different fields and communities, and we'll talk a little bit about what they mean. Very often, these interactions either change state or transport things. So if I have an arrow that represents the blood vessels that connect, say, the liver, the gut to the liver, then the arrow would transport the chemicals in the blood from the gut, absorbed in the gut, to the liver. And so it would give me a rate of transport. If the arrow represents airplanes flying from city 1 to city 2, the arrow could be the number of people moving from city 1 to city 2 per unit of time. The arrow could also change the nature of something. If I cough on somebody and they go from uninfected to infected, then I'm not changing the amount of something, I'm changing the nature of it. So those are called existential processes. And there's a whole rather detailed and initially not that critical for our purposes literature about how you describe these various kinds of change and interaction. That's actually pretty profound, and we actually have people here who specialize in that. It's Bandiar here at IU is an expert on those kinds of things. But those kinds of issues, until you've worked with networks and understand why those are important questions, they tend to be pretty dry. Once you've dealt with networks, you understand why people spend their lives addressing them until you run into them a little bit of time. Okay. A warning, and I'll try to use red for warnings or blue tends to mean something about important, sort of an underline in my slides. Red means watch out. So different subject areas and even different individuals with the same subject area tend to use different ways of drawing things. Networks are very visual. We lay out nodes and links and arrows, but not everybody uses the same nodes, links, and arrows. And for example, in economics, very often the arrows actually represent things and the nodes represent links. Why economists do it backwards, I can't tell you, but if you look in economics papers, they very often flip that. And so most of the places that we are going to deal with, the nodes are going to be things and the arrows are going to be interactions. But I can't promise that's 100%. If you pull an interesting paper off the net, somebody might do it differently. So again, remember that the network is a very general concept. And so you could have different numbers of nodes, different kinds of nodes, different states living in nodes. You could have substructures of nodes. You could have one kind of link or multiple kinds of link in the same model. And I mentioned already directed and undirected nodes, bidirectional nodes, nodes that can only go between links of particular kinds. In biology, we'll also see something pretty often, which is arrows that point not to nodes, but to other arrows. And those are called modifier links or regulators. And we're going to have to understand how to deal with those at some point. In general, I started out saying that links connect nodes. And now I'm pointing out that in biology, you're going to run into links that connect links, well, or nodes to links. So we're going to have to be ready for that. Now, it's a little bit of introduction to networks. But now we've said that this class is about the dynamics of networks. What are network dynamics? But I said before that there's a whole field of networks. The Network Science Institute people do a lot of this, which people like Connie Berner do a lot of this, which is focused on network architecture. What is the shape of the network? How many nodes? How many nodes connect to how many other nodes? What is the statistical distribution of connections? But in this class, we're going to focus on how networks change in time, dynamics. And that's, again, because we want to be able to control things. And if we're going to be able to control things, we have to change them. And there's a nice paper, online paper, which had some beautiful pictures, which I thought was a very good example of this, which is that networks can change in two distinct ways, or both at the same time. You can change network architecture, that is the identity of the nodes or their number and the connections between them could change. And that would be the middle one here. Here you see I have the same number of nodes, 5 of them, but the links between them are different before and after. I've added some links and I've taken some links away. And that's evolution of network structure. Another thing would be to say that the network architecture stays the same. That's the left-hand one, but the state of the node changes. And that's the thing we're going to be focusing on primarily in this class. Dynamics on networks, not dynamics of networks. And the reason we're going to focus on that is that dynamics on networks is described in terms of continuous processes. In a dynamics on network situation, I could change, say, the amount of glucose in my cell. I could change the metabolic state of my cell. I could change the amount of virus in my lungs. Those can change continuously. But in a dynamics of networks, when I add a link or subtract a link, that's a Boolean operator. I either have the link or I don't. And in principle, you have what they call here adaptive networks, where you could actually change both things at the same time. The modeling methodology that we're going to use in this class is designed for modeling dynamics on networks. It's not designed so much for rebuilding networks and changing network structure dynamic. There are software packages, and there are certainly ways you can do dynamics of networks computationally, but they tend to be rather specialized and they're harder to learn. And so we're going to focus on dynamics on networks. It's actually possible, for example, in neural networks, to sort of have the best of both worlds. It's just computationally inefficient, which is if you have all of your nodes and you have the potential to connect every node to every other node, what's called an all-to-all network, and then you assign a strength of 0 to the ones you're not using, then you can have dynamics of networks implemented as dynamics on networks. We probably won't deal with that so much in this class, but it's something to be aware of. Okay, so we're going to be focusing on dynamics on networks. That is, we're going to draw a network architecture. We're going to define what the states are. We're going to ask how they change in time. Right. Again, the state that we're interested in is going to depend on what the problem is. So it could be the amount of chemical, like ATP, retinoic acid, fibroblast growth factor. It could be the number of dollars in a bank account. It could be a state question, whether a person has the flu or not, number of airplanes in a city at a given time, population of the city, how fast the neuron is firing, differentiation status cell time. And the first homework problem is going to ask you to think of a network and describe, just verbally describe how it works. And I encourage you to be creative. I show that United Airlines one all the time, and then I get 10 homework assignments, all of which are United Airlines flight map. It's okay, but you try to pick something that's interesting. Okay, again, the links represent interactions which change the node state. Okay. So let's think about applications of networks. I've already mentioned the example of economic activity and trade that are often described as networks, production networks and trade networks. Trade networks, classic use of it. Neural activity in the brain, neural networks. Electrical charge on membrane may be less obvious as a network. Epidemics, clearly if it still says influenza, it's an old slide written before COVID. Animal populations in an ecosystem, so eco networks. Pollutant flows in the city. I put a toxicant into the groundwater and it gets carried around. Or my car belches exhaust. Transport of blood, chemicals within the body, we talked about. Gene regulatory networks, we talked about. Chemical reaction and so on. Almost all of these are also things that do have a spatial component. The network models typically leave out the spatial component. And again, I mentioned that in the spring, I teach a class where we put the spatial components back in. But one of the questions you might want to ask yourself when you're building models of networks is it legitimate to leave out all the things that you're leaving out when you build a network model? Okay. So again, coming back to networks, every dynamic network has three things. And no exceptions to that. There are very few things in this class that don't have exceptions, but this one doesn't. You have nodes, which have states, which are variables. You have links that define which nodes affect the states of which other nodes. And on each link, you have a rate law that describes how the state of one node at one end of the link changes the state at the other end of the link. And that basic set of nodes, connections, and rate laws on the links is the fundamental thing that we're going to deal with again and again and again in this class. Mathematical formulation is going to be the same, whether those links are representing dollars in the bank or number of people infected in the population. However, the specific form of the rate law on the link will be different depending on what it is you're trying to model. There are also some other things that you have to think about, which we'll deal with a little bit in the class. One of which is that rates of change can be continuous or discrete, and they can be deterministic or stochastic. Let me give just one example. A continuous and deterministic example would be, suppose that I have a pipe that's carrying water, and my nodes are a bucket, which is a source of water, and another bucket, which is being filled. The amount of water at the one end is changing, going down, and the amount of water at the other end is going up. It's changing continuously, and the flow rate is going to depend on the resistance of the pipe and the pressure difference at the end. It's deterministic. On the other hand, if I ask a question of, I'm at the bank waiting to see a bank teller, and there are people ahead of me who are being served, and I want to know how long the line of people is in waiting to go to the bank teller. People come in, they're discrete. The bank teller doesn't see a half a person at a time. He can only see them in units of one. The amount of time each transaction takes is going to depend on what they're doing. So there's some randomness there. And the rate at which people arrive is going to be random. And so that's a discrete problem, and it's also a stochastic problem. Molecular interactions in biology are sort of an interesting mixture of those two things, because at the level of single atoms and molecules, single molecular interactions are discrete, and they're also stochastic. They're probabilistic. The rate that whether two molecules react or not is a stochastic process. On the other hand, if you have a lot of molecules, we can write rate laws that say if we combine two chemicals together, the rate at which they react is such and such. And so very often we will take what's called a continuum approximation, where we assume that a fundamentally discrete process is continuous. And we'll do that with populations as well. When we model infection, we're going to assume that the population is big enough that we can treat it as a continuous variable, even though in fact it's made up of individuals who either are infected or aren't infected. People are either alive or dead. It's in the mix, but we're going to assume that there is. And one of the things that we'll explore relatively early in the class, and tellurium allows you to do pretty easily, is to switch from the continuum approximation to the stochastic one. A single line of code can do both, and you can see whether they're giving you the same result. Okay, so how are we doing here? Classically, this class would have a break in the middle, but I'm not supposed to do that because of the COVID restrictions. Normally, I would bring food and we would have a snack break in the middle of class, and I can't do it. I'm sorry about that. So let's go ahead. Let us actually now build a simple model together. And we're going to use tellurium, and we're going to use it on the computer. And I'm going to have to figure out how to do this effectively here in the classroom. Normally, if I were teaching this online with the Zoom, I would have a split screen where I would have the tellurium on one side and the lecture slides on the other. So we'll have to figure out. I think I could get that to work here. So we're going to start doing epidemiology, and we're going to do the classic, gradually build up to the simple, very simple model. We're not going to probably get there today, next Tuesday, of what are called SIR models: susceptible, infected, recovered, or resistant. And until 2 years ago, most people in the world probably hadn't heard of epidemiological models. These days, epidemiological models are in the news all the time. R0 or R0, the infectious rate of the virus, is something that's in the news. Is it bigger than 1 or less than 1? People want to know. R0 is not a property of a virus. It's a property of a model. And we'll find that out pretty quickly. When we look at viral epidemics, we see that there's a typical pattern. And this is infections in Italy early on in the epidemic last year, in February and March through August of 2020. And what you see is that you get a very rapid or early growth of infection. May or may not be exponential, or it looks typically exponential. You reach a peak, and then you have a slower decay back to some low baseline. If you integrate that, this is the death rates, the total deaths in England over that same period as here, Italy. You see that the number of deaths is very low initially, it increases, it reaches a maximum, and it slows down. And back a year ago, when I was teaching this class, people sort of hoped that that was how things were going to work out. In fact, if you go to Worldometer and you look at new daily infections in the world, what you'll see are what are called waves. That first wave, which is the one up there, the things went down. The second wave, third and fourth wave probably together, and then some people would say that's third, and then fourth, and now we're in the middle of the fifth wave. Sorry?
What is disease
COVID.
for? Oh,
These are all COVID. If we look at established diseases like influenza, we see not totally different pattern. What you would see for influenza is that there are 2 peaks of infection per year. There's a peak every fall, mainly associated with people going back to school, and a peak in the spring, with a dip in the middle of the year and a dip in the summer. Influenza also has a complicated spatial pattern because it tends to be associated with cooler weather. And so influenza rates tend to be high in the winter, the fall, and the spring in the northern hemisphere, and then they flip. So because they're actually a little late. So late fall, early spring, that is sort of early winter, late winter peaks. In the northern hemisphere and the southern hemisphere alternate. And so you see influenza rates are high in Australia when they're low in the United States. They're low in Australia when they're high in the United States. But COVID doesn't seem to follow that pattern. There's been a lot of debate about whether how much the weather affects COVID. If you look at this picture here, the lifetime of each one of these waves is about 4 months. Again, it's not quite understood why that is. This is worldwide, and of course, different countries have had very different trajectories. India had this huge peak, and then a very rapid decline in cases. Now it's getting a second peak. The US actually more or less matches that pattern with bigger dips in the middle. So I strongly recommend Worldometer. It's a great source of data. It's a great source of things we can model. There are a couple of caveats which we'll come to, because there are a couple of things that are not in the Worldometer data that are not maybe what you expect them to be. Another thing that we could look at is the dynamics of viral infection within a host. So instead of now, somebody says, can you not hear me anymore? Did you lose? Okay, thank you. Sorry, guys. Thank you for, I do have the chat window open. So if you type into the chat window, I do see it. Okay. So one of the things that we can ask is, now somebody gets a virus particle in their lung. That virus infects a cell. That cell produces virus and infects more cells. That's actually very similar to the process where one infected person infects a bunch of people in a population. They go on to infect more people and so on. Just the way in the population, individuals typically clear the virus, unless it's something like HIV or tuberculosis or bacteria, where you typically carry it for a long time. Hepatitis, another disease you typically carry for a long time. In the case of cells, things are a little bit grimmer, in the sense that almost never, and there always are exceptions in biology, but for most of the viruses that we deal with, once a cell is infected with a virus, the only way it can get rid of the virus is to die. So once a cell in your lungs is infected, it will die from the virus or from your immune system. And so in this case, you have a bunch of uninfected cells here, which are reproducing themselves. They encounter the virus, so they become infected. Typically, people may have heard the term eclipse phase or latent phase for virus, where the cells are infected but not producing virus. At the population level, that would correspond to somebody who has been infected but is not yet infectious. In epidemiology, you also have to worry about the fact that you could be infectious and asymptomatic, which is when you tend to spread it, because once you get really sick, you don't go around spreading it. So this virus cell is infected but not producing virus. It then goes to a situation where it's infected and releasing virus. And that virus is available to infect new cells, and that cell will die. And some of those deaths are due to the virus itself, some of those deaths are due to the immune response. If I look at the different kinds of pattern of infection within your lung, for example, and ask what the viral load is, as I mentioned, the experimental data, clinical data on this are not that great. Most of this kind of data comes from in vitro experiments where you infect cells in a dish, which aren't quite the same. You see that there are different typical patterns of infection. A classic what's called monophasic situation, the amount of virus in your body goes up very rapidly, reaches a peak, and then comes down very rapidly. Another situation is it goes up very rapidly, then comes down slowly at first, and then clears very rapidly after a certain delay. And that's typical for influenza. The amount of influenza in your body goes up by a factor of 1,000 per day once you're infected with flu. You don't start feeling sick till day 2, the end of day 2, which means you've had 48 hours to go around infecting lots of other people. Actually, typically, by the time you start feeling sick with the flu, the amount of influenza in your body is already going down. You don't even know you're infected until after the peak of the flu. The reason you feel sick is because your immune system is kicking in to get rid of the flu. One of the reasons people think sometimes that when they get a flu shot, that the shot is causing them to have the flu, is because the symptoms we associate with the flu are the immune response to the flu, not the flu itself. Of course, if you didn't have the immune response to the flu, you'd die. So that would be different. You would get sick. It would be a different matter. And then there are other patterns. You could have a situation in which things go up, peak, then go down slowly, and stay on some plateau for a long time and then clear. That's more like COVID. If you're unlucky, it can peak again, and then you can have real problems. And there could be a situation where you never clear it, where you live with it. HIV would be an example of that. And so there are these different patterns of infection that you see. And you'd like to be able to understand what causes these different patterns of infection. Your immune system is the same. The basic way viruses replicate is pretty much the same. It's more about the details of the parameters in the model that make those different. And there are different levels and types of models of viral infection within bodies. There's this nice paper, recent paper of Erica Mohan and Bart Ermintrout and TJ trying to model viral infection in the lungs, understand those different patterns. That would actually be a really good paper to replicate. There are very complex models like that one on the top, which is also of Erica and Bard, which model in detail lots of the different kinds of immune cells, lots of the different chemicals that the immune system uses to signal the cytokines and chemokines. There are also these very simple models. Alan Perlson at Los Alamos National Labs is one of the great pioneers of this kind of modeling. He worked very, very much on HIV. And a lot of modern HIV therapy is derived from the work he did modeling HIV. He's also worked on hepatitis C and a lot of other diseases. He likes very, very simple models. He does minimalistic models. And then there are other models. This is another Alan Perlson model, in this case of influenza, a little bit more detail. So there are all sorts of different models of viral infection within the body as networks. Some of them are very simple. Some of them have many molecular and cellular components. Some of them treat the whole body as one big blob, stirred reactor. Some of them would model the upper respiratory system, the middle respiratory system, and the lungs, lower, the alveoli and the lungs separately. Some of them might ask the questions about how the disease goes from the lungs to the bloodstream, for example, which is important in important in the pathology of COVID. So now let's come to the very simplest model that we're going to work with, the SIR model. I mentioned it by name before. This is a susceptible, infected, and recovered model of epidemics. It's also very similar to what we're going to do in the host. So we are going to think about a situation in which people only have 3 states. And we're going to have 3 nodes in our situation. We're going to have the number of people who are uninfected and who could be infected, S. The number of people who are actively infected, I. And the number of people who are recovered. And we're going to assume here, which is unfortunately not true for COVID, that the recovered people can't be reinfected. Now, in some cases, R is a euphemism for dead. And if you're dead, you definitely can't be infected. Although you can be infectious. I mean, unfortunately, last year, some people worked on Ebola, and you're actually more infectious after you die with Ebola than you are when you're alive. At least until your body is disposed of. So we're going to build this very simple model. And this is, I mean, almost doesn't deserve to be called a network, but it does have 3 nodes, S, I, and R, and 2 arrows. S goes to I goes to R. And the state is going to be the number of individuals in each category. If we think about this as a chemical reaction, it conserves mass because the total number of individuals in the model is conserved. We don't have people being born, we don't have them dying. If we had people being born and dying, you'd have extra arrows coming in and out of this. Okay. And this arrow diagram actually has the form of a chemical reaction, a very simple chemical reaction. S turns into I turns into R. Okay. So going back to our arrow diagrams. This is going to be just a little bit of an analogy between chemistry and the kinds of models we're going to do today. Again, our nodes represent states, and our nodes represent different populations, and the state is going to be the number or the density of those populations. We're going to typically use plain Roman, and occasionally PowerPoint will change something to italics when I don't want it. So I can't promise if it's going to be right, but for the names of things, S, I, and R. Links with arrows are going to represent transformations. Chemically, that would go from substrate, that's an input, to a product, that's an output. And typically in the world we're going to live in, links are going to be unidirectional, although in chemical reactions, you can have 2 molecules of ADP go to 1 molecule of ATP plus 1 molecule of AMP and backwards. All right. So this gives us a network architecture. S goes to I goes to R gives us a network architecture. But we want to know how S, I, and R change in time. And to do that, we're going to have to go through a series of steps. And the key thing that we're going to have to do is write a rate at which S, the susceptible people, become infected, and a rate at which the infected people recover. And so we're going to always go through the same sets of steps in analyzing this problem. The first step is we name the population. So S, I, and R are going to be uninfected people, infected people, and recovered people. And the population of infected, uninfected people is going to be written with an italic S. The population of infected people is going to be the italic I. The infected number of recovered people will be written with an italic R. So we're going to be distinguishing the name of the node from the state of the node with Roman and italic. Okay, the next thing we do is we have to say that there is some rate which converts S to I and I to R. What that rate is isn't specified, but there is some rate. And I typically write that by decorating the arrow with a little lowercase italic letter that defines the rate. It's often known as a velocity, which is why it's using V and W. And the next thing that I have to do is say what that rate depends on. And that's the critical thing. And typically, in these cases, the only thing that that rate could depend on are the things at either end of the arrow. Very often only at the rate that depends only on the rate, the thing at the source end of the arrow. And so here, the velocity at which I convert S to I is the function of the amount of S and the amount of I. The velocity at which I convert I to R is the function of the amount of I and the amount of R. Now, in reality, there could be additional arrows that change those rates. But we're going to stick with that for the moment. To begin with, we're even going to simplify beyond that and just look at the process of infection. S goes to I. The actual rate at which people get infected in the population could be very complicated. It could depend on all sorts of things besides how many people are currently infected and how many people are uninfected. If you're wearing a mask or not wearing a mask, that'll make a difference. If you go around when you're very sick or not sick, that'll make a difference. If you're vaccinated or not vaccinated, that'll make a difference. None of that is in this model. All we have are two populations, S and I. And so we don't have anything else we could put in the model to use. That means the model is not going to be very effective at actually reproducing reality. The other thing we're going to have to assume is that we're going to build a model to begin with using a continuous approximation. We know that individuals come in integer units. You can't have a half a person. We're going to assume that their population is big enough, 1 million people, so that we can approximate adding or taking out one person as a continuous process. We're also going to assume that the rate of change is deterministic, even though actually each interaction event, each infection event is stochastic. And so we're going to neglect the stochasticity to begin with and the discrete nature of the population. Those are both big assumptions, and especially early in an epidemic, they're probably not right. If you have millions of people infected, it's a reasonable approximation. If you have only a few, it's not. And the next thing we're going to assume, which is another big approximation, is that the infection rate is uniform over the population. And that's clearly not true. Some people don't go out. Some people mix in large crowds. And so that assumption that the rate of infection is uniform is not really a very good one, but we're going to make. Another thing that we're going to assume is that every individual is equally likely to be infected, which is also not true. Some people may have natural immunity. Some people may have impaired immunity. Some people may have been exposed to the virus before. And so those assumptions are all pretty harsh ones. And it's actually a little bit of a surprise that they work, but they do pretty well. If you could make all of those assumptions, then you can write those rates in the form of simple first order ordinary differential equations. And those are called reaction kinetic equations. And so typically this arrow diagram, S goes to I goes to R. If the rate S goes to I is VF, the rate I goes to R is WF. Then I could say the rate of change of S with respect to time. There's no source for S, so it's minus VF. The rate of change of I is the amount of S turning into I, VF, minus the amount of I turning into R, minus VF, WF, sorry. And the amount of R being produced is just WF. And so now we've converted a picture, a diagram, network architecture, into a set of differential equations. And in this class, we're going to use a little calculus, but the calculus is never going to get harder than first order or time rate. Okay, now if we actually want to say something definitive, we have to know what V and W are. Once we define those functions, we've defined the problem. And the very simplest expression we can have, and this would be true for population or for cells in a dish, is let's call that initial state, I called it S before, we'll call it T now. And we'll say that we have cells T1, there's a population of uninfected individuals, and the number of infected individuals is I1. And now let's suppose that the rate of infection for time is the amount of virus in the population, V, times some infectivity of the virus, beta. Now, in reality, that virus has to be produced by something. And so the amount of virus would really depend on the number of infected individuals. But to begin with, we're going to assume that the amount of virus is just out there somehow. And we're going to say then that the rate at which we convert T to I1 is proportional to beta, the rate of infection, times the amount of virus, V, times the population, T. Why do you have T? Well, suppose that I didn't have T. Suppose the rate of transformation from T to I1 were just beta times V. If I do that, what's going to happen to T after a while? If T goes to I1 at a rate beta V, what is the functional form of T as a function of time? Beta times V is a constant. So I'm decreasing T at a constant rate. Well, T minus T0, the initial amount of T, minus beta V times time. Yeah. Okay, that goes negative. But my population can go to 0, but it can't go negative. And so multiplying by the number of people who are available to be infected limits me on the negative side. So again, if I've written the rate law, beta TV, dt, and I'm sorry, because of the t's on both sides, is equal to minus beta times the conceptual population times the virus. Number of infected is the reverse of it. If I add them together, I conserve the total number of people or cells in the population. All right. This one I can solve analytically. dt by dt, d big T by d little t equals minus beta. Yes, go ahead. About the rate? Well, so the assumption is that the probability for each individual is getting infected depends on beta and V, and it's independent for each individual. So the probability for each person independently is beta V. And so the total probability, the total rate is the sum over all individuals. That's T times beta times. Thank you. Okay, so we're going to start out with T of 0, the number of uninfected individuals at the beginning. A lot of times the amount at time 0 will be written T sub 0. That's a notational standard. And we'll assume that initially there are no infected individuals. And if we plug that in, it's probably the very first thing anybody learns in the calculus course. You get a decaying exponential. And that's something, there are not very many things that people need to memorize, but not recognizing that first order decay gives you an exponential is something that you need to learn. If that's unfamiliar, I recommend looking at a basic calculus. We're not going to use a lot of calculus, but that level. Okay. So in this case, the number of uninfected individuals per time is the initial number times the exponential of minus beta v times the time. So we solved the problem exactly. You don't need to simulate it to know what's going on. But let's actually do it as an exercise. And that will be our first little project in class. In general, after today, I will try to minimize the amount of time I spend talking all simultaneously and do more in-class exercises. We'll have a few minutes of discussion and then we'll try something. I'll have a few minutes of discussion. The first class always is lecture heavy. Okay, so we are going to use Tellurium on NanoHub. We are going to be using the Tellurium app on NanoHub. It runs a simulator called LibRoadrunner, which is written by Andy Smudgy. As I say, you can do it locally, but we're going to do it online. And it will look different and may behave slightly different in terms of the way plots work. So I want everybody to create a NanoHub account. How many people here in this room have a NanoHub account? 1, 2, 3, everybody. Good. How about online? If anybody doesn't have a NanoHub account, please type it in the text so that we had the chat so I know we need to help you out. Wonderful. Everybody has a NanoHub account. Okay, so I would like people to fire up NanoHub. Does everybody know how to get into NanoHub? Once you have gotten to NanoHub, log in. Okay, somebody says they just made one. All right. All right. Setting up the account takes only a minute or two if you're at IU. Okay, sign in. Go to the search bar, type in Tellurium. And let me see if I can set up a split screen to do this on this computer. Having the mouse go backwards is confusing. All right. I will reshare the screen just a second. Let me just get the split screen set up. Oops. Okay. Okay, can people see the slides and the tellurium window? So please find tellurium. And once you find tellurium, you want to go to the tellurium web tool and you want to hit launch tool. Okay. And I will use polls for people who are not here. I will do what I will typically do is I will run a poll which asks you, how are you doing? And you'll get a poll that says, need more time, finished with exercise, and so on. One problem with polls in Zoom is that you can't change your answer. And so if you, I would encourage you not to, if you need it, in general, it's fine to say, if you say need more time, then I'll poll you again. But typically, don't answer, I need more time until it looks like I'm about to go on to the next thing. Okay. And if you need help, click need help, and then we'll do our best to help out. So is it clear? Does everybody understand how to get, how to launch Delirium? Anybody need help with that? I'll just try launching the poll again. How about in the classroom? Does everybody have it? Okay, so now, once you're in Tellurium, for today, we probably don't need to do this. There's a minor annoyance with the way Tellurium is configured. When Tellurium is launched initially, it is a read-only notebook. And so my recommendation is to copy the import Tellurium as TE, and then you go to, you have to go to the file. Why is this showing? Huh. The menu options are different from usual. Save and check file. Oh, I see. Because of the size it's gotten. So the display is a little bit different because the size is different. Okay, if you go to the file menu here and click new notebook, it'll then give you an editable notebook. You have to repaste in your import Tellurium. Again, the way this is displaying right now, it's not actually showing it. But if you see this note at the top where it says read only here at the top, you may have to restart it. If you do that, you have to retype import tellurium as TE. In general, we'll be using NumPy a lot, so I recommend typing import NumPy as NP. But today we probably won't use it, but it's something that's helpful. The next thing that we need to know how to do is how to do uploads. To do uploads, we need to, I don't know what, I'm sorry. Something is funny about the way Tellurium is configured here. To do uploads, you go to the File menu. You hit Open. And you hit Upload. And I'm not going to go through that. You can do that if you like. The student materials folder has a copy of the simulation we're going to run now. So let me give everybody the link to that. Sorry about that. Zoom and this display aren't interacting in a friendly way. Okay. There's a link to the folder you should be able to get into. We'll do an upload. And we will go to where is the student materials, lecture 1 demos, simple viral infection 1. Right. Once you've selected that, you have to hit upload. And it should upload. And we'll have, if this doesn't work for you today, we can type everything ourselves because it's not going to be that much to type. But you have to hit the upload box to do the upload. And now we click on the model here. And we should have the model. Were people able to get that to work? Yes, no, maybe. Yes, yes. Yeah, you need, sorry, I should have said that. You need to download it. The other thing is, I think it should be on Canvas. And for next time, we'll make sure that I will ask people to do the download first. I'll try to have the demos all uploaded early enough. Sorry? The file should be called Simple Model of Viral Infection Lecture 1, Exercise 1. And depending on how your computer is configured, you ought to be able, if you have Google Drive running on your computer, that link, you should be able to upload it directly from that link without having to download it to your computer locally. If you don't have, if you're not running Google Drive, you probably have to download it. Again, I'm having to get used to the layout of things on this configuration here. It's not set up the way I'm used to. Are people able to find it? Are you able to download it okay? Yeah, that's right. It should open. Did it, how are people doing? Let me pull my online. Okay, does anybody, are you able to get it? Upload is, you go to file. So for some reason, it's displaying slightly differently, probably because the screen is so small. If there should be if you go to, if you have the standard layout, it's under. So you go to file, open, file, open. So if you look in the user interface at the top left, there should be a file pull-down menu. For some reason here in my display, it's folded all of those pull-down menus into that menu option at the top. So it's not displaying it quite the same way. It made a liar of me. I told you that if I used it online, everyone would see the same thing, and there it's not. So that's life, I guess. Okay, and so you go to open, select file, then you have to find your file, set upload, and then once you've selected upload, you go to hit upload, and then you point to the file which you want to upload, and then you have to hit upload a second time. Okay, are you able to get it? Great. Okay, we're going to do that a lot, so we'll get used to it. Okay, and then once you got it, you hit simple model viral infection, and it should pop up Jupyter Notebook with the thing in it. In this case, it's run it. It's already run it. And this is a slightly decorated, it's not just the basic thing because it actually has some text explaining it. But we have our model. And if we want to run the model, we click on the window and we go to run and we hit run. And it'll run it and it'll display the output at the bottom. You can also go into the window and you can hit Shift, Enter, and that'll run the cell. So if you hit Shift, Enter, that runs the individual cells that you're in. How many people in the room have used Jupyter Notebooks before? Everybody, almost everybody. 4 out of 5. Okay. Have you used Python before? Okay, but a different Spider or something, different IDE. They're all similar but not identical, so there's always a little bit of and the version here on Jupyter Notebooks is on NanoHub is slightly different from the standard. Not a lot. Were you able to get it to run? Great. Okay. Good. Okay. So when we want to save things, there is this issue that if it's read only, we can't save it. So we have to restart once the file. For today, we're going to ignore that because we're not going to save it. I'll just tell you how to save things for reference. You click on the file option. Once you click on the file option, you can do rename. Give your notebook a name, because if you start out typing a new notebook, it won't have a name. Then you hit save and checkpoint. And once you've hit save and checkpoint, it will give you an option to download as, and you can download as a notebook. If you want to run your application locally and you're not using Jupyter Notebooks locally, you'd select download as Python, not download as a notebook. Now, in principle, NanoHub saves the stuff you've got. You've got a local directory space and your files should be there. If it's important to you, I really recommend downloading, because NanoHub does occasionally clears the working directories. And I don't know quite what their rules are. The stuff I have has been sitting there for years, but I know students occasionally have had issues where they saved something locally and it disappeared. So, for the little exercises we do in class together, it's probably not a big deal, but I recommend anything you're doing that you want to keep, download a copy. Okay, so let me explain what this code is doing. So the first line of the code, and we're going to see this all the time, is, let's just talk about Jupyter Notebooks a little bit and how it works. So Jupyter Notebooks is a notebook like Mathematica or MATLAB. The boxes are interactive cells. And so in each box, you can type text or Python or other things. And if you want to evaluate a cell, you hit Shift Enter, or you can go up to kernel. And under kernel, you have an option of running all of the cells, only one cell, the cells below the cell you're in, and so on. Or you can hit Run here. So you've got a lot of ways of executing the code. And the first thing that we're going to do always is import Tellurium as TE, because Tellurium is the library we're using. Okay, then we have actually the code of the exercise, which is here. And I want to walk you through, before the rest of the class, I want to walk you through this little code and explain what it's doing. As a reference, there is that one-page guide to Tellurium that I printed out and that I also sent out as a PDF. There's also this great Tellurium readthedocs.io online manual, which I also strongly recommend. Depending on how big your computer is, this is another reason perhaps it's easier to do this at home. If you have two video displays, if you're at home, your laptop and a secondary display, I would recommend on their secondary display keeping the manuals open. Or maybe having the videos from the class and then the right, Tellurium. So let's actually look at this model definition, because this model is implementing this very simple simulation. So the first line is import Tellurium as TE. That's telling us that we're going to be using Tellurium as our modeling language. The next thing that we see is something that says model string equals, and then a triple quote. And our actual model is going to be a string, which is the model definition. And if you're used to procedural languages, this works a little bit differently. I'm not going to go into too much detail on what those differences are right now, but I want to just let you know that you're going to find that things are not quite perhaps the way you're used to with procedural language. For people who are not Pythonic, the triple quotes allow you to have strings which have character terms. That's why I use triple quotes. Okay. And then if I go down to the bottom, I see some actual Python. M equals te.load A. And we'll come back to that. That is taking that string and converting it into Python. S equals m.simulate. It's actually running that model that was specified in the string. And m.plot, which is the method that puts the results of that simulation up on the screen. So let's walk through this code line by line. Again, import tellurium TE imports the methods we're going to be using, and we're always going to have that. We don't have to use the abbreviation TE, but that's the standard abbreviation. Just the way NumPy gets abbreviated NP. The second thing is that our model definition is actually a character string. And so we have model string equals open triple quotes with a pair of closed triple quotes at the end. And then here in yellow is actually the model definition. You're going to find some differences between Epidemiology, the language we were using to define the model, and Python. And so all of our models are actually going to be using two languages, one for the model definition and one for the model execution. That would be more of a pain if the model definition weren't so simple. Because having to learn two languages at once would be a pain. We will find that there are a couple of things that are different. For example, double slash is the comment character in Epidemiology, whereas hash is the comment character in Python. Python cares a lot about indentation. Everything depends on indentation in Python. Epidemiology, you don't have to indent, although we typically will indent. Semicolon is a separator, like in C++, in Epidemiology. It's equivalent to a line break. Okay. And so now we're actually going to write the model, and this model is a simple model. T goes to I1 at a rate beta times T times V. And it's written exactly the way it looks. T arrow I1, T arrow I1. And the rate law is written semicolon beta times T times V, close semicolon. That E1 colon is naming the reaction. We don't need it. We could leave that out. But it's good form to name your reaction. It's like naming equation 1 in your paper. Okay. So if I see an arrow diagram, T goes to I1, I write T arrow I1, semicolon, whatever's sitting on top of the arrow, I type at the end of the line. And that's about all there is to writing Epidemiology models. I mean, we'll find there are all sorts of bells and whistles, but 98% of what we need is that. And that's why I like to use it, because it's really pretty simple. Okay, now I have to, okay, so here's that, that reaction specification. If you're used to writing equations using something like MATLAB, where you write the ordinary differential equations, you have to get used to the fact that an arrow diagram actually creates two differential equations, two rate laws, one for the species T on one side of the arrow and one for the species I1 on the other side of the arrow. So writing this one line creates two differential equations. Again, walking through this, E1 gives a name to the reaction. And I can actually reference the rate if I want. If I ask for the value of E1, it'll tell me what the rate is. T goes to I1, who's defining the model structure. It says that those two nodes, T and I1, have that relationship. And beta times T times V tells me what that rate law is. I could have some other rate law, beta times T squared divided by V. I can write anything I want, anything I can write arithmetic. If I had a more complex reaction, I would have more lines. But each line is going to look like that. Okay. We're not going to be using it so much here, but if we were writing chemical reactions can be rather more complicated than what we have here. A chemical reaction might say 3 molecules of A and 5 molecules of B go to 1 molecule of A, 2 molecules of C, and 9 molecules of D. If I wrote that as differential equations, it would be quite a bit. That's one line in antimony. 3 times A, 3A plus 5B goes to A plus 2C plus 9D, and then the rate. And antimony will automatically take care of subtracting 3 A's for every 5 B's and so on. That's called stoichiometry. And that means that when you're writing complex chemical reactions, they're extremely compact using this formalism. And antimony was originally developed to write chemical reactions. It's sort of an accident that we can write all these other things too. So that one line there would actually generate these 4 ordinary differential equations. So if you're used to MATLAB, antimony is a lot more compact in modeling. Okay. There are a couple of other things that we're not going to use today, but I want to introduce as concepts. Arrows don't have to, sometimes you don't care where something comes from. If children are being born and they're susceptible, and they're being born at a rate K, that's called a source term. You have an arrow goes to A coming from nothing at a rate K. That's equivalent actually to writing an ordinary differential equation explicitly. Arrow goes to A is equivalent to writing dA by dt equals whatever the rate law is. But if I actually wanted to write explicit ODEs, I could write them that way. Arrow goes to whatever my species is. If something disappears and I don't care what happens to it, people die and I don't care about what happens, so I don't count them. I can also have A goes to nothing. And A goes to nothing is dA by dt equals minus Ka, in this case, rate K. And that dissolution term or degradation term is something we'll use pretty often. There's a typo on that slide. All right. We can also write an ODE directly using a apostrophe. If I write T prime equals beta times T times V, that gives me the single differential equation, dt by dt, d big T by d little t equals beta TV. T prime is equivalent to writing arrow goes to T with nothing on the left-hand side. typically we'll avoid writing ODE style, but we can. Another thing to be aware of is that if I write multiple reactions involving the same species, they get summed up automatically. If I write nothing goes to T at a rate K0, T plus V goes to I1 at a rate K1 times T times V, I1 goes to D at a rate K2 times I1, then my T has a term from E1 and a term from E2. And so I'll get two terms in my T equation. My I1 has one term here from E2 and one term from E3. So I'll get two terms from I1. And so if I write multiple lines of reaction, I have to sum when I convert them to ordinary differential equations. Well, because we're thinking about these as pools, if some process is creating infected people and some process is getting rid of them, the net rate of change of the infecteds is the sum of those two rates. And so if I don't want them to be summed, I should really call one of them T1 and one T2 to distinguish the population. So typically this is convenient rather than a problem. The time that you have to think about it is if you are trying to read a paper that's written things in the ODE form, and you want to know how to write them in tellurium, you have to take the terms for all the terms that involve T and combine them and pull them out. And we'll practice that. It's not something you learn instantly, but after a couple of exercises, you'll get used to it. Okay. The next thing we have to do is we have to specify all the values. If we have a parameter beta, the infectivity of the virus, we have to give it a numerical value. And it's good practice to define as a comment the units, what the units of that are. If there's an amount of virus, we have to say how much there is. And we have to give the initial values for all of our chemical arbolar species, with the caveat that a variable that's not named gets a value of 0. And so if I don't name I1, if I don't give I1 an initial value, it'll be assigned 0. And that's actually bad practice. This simulation is not as pretty as it should be, because in fact I should have written also the line I1 equals 0. If you look at my initial conditions, I've done something else, which is I've used a variable. I've set T0 equal 1e to the 7th, and then I set T equal to T0. Now that looks pretty normal by programming language standards. We will actually find that there are some complexities when you go to doing multiple assignment in tellurium, which I don't want to worry about right at the moment. The language antimony allows you to do a lot of other things. We can define piecewise linear functions. We can define predefined functions. We can define events, things that happen at discrete times or under particular conditions. For this model, we don't need those. We will use them later on. One of the things that I don't want to get into too much today, but we'll deal with next time, is that antimony is what is called the declarative language. So in a language like Python, which is a procedural language, it's object-oriented, but procedural, statements are executed sequentially. And so there's a concept of the transfer of flow from statement to the statement to statement to statement to statement. In a declarative language like antimony, things typically are understood to happen simultaneously. If I write 4 ordinary differential equations, those ordinary differential equations proceed in time at the same time. And so all of the rate laws that I define are conceived of as happening sequentially in time, I mean, simultaneously in time rather than sequentially. The one exception to that is in definition of initial conditions, where initial conditions are evaluated once and only once in the order in which they're written. Let me finish because we're running out of time. Just walk through the last couple of lines here. Once we've got our model definition, we have to load it, and we create an object, a reference to this object. The method TE_load_A takes that string and converts it into an executable. So it actually takes the string, it compiles it into C++, and then it's going to execute it. And we're never going to play with that. We're just going to assume there's a parser that does its job. So the Python object M is an executable. And then the tellurium method simulate takes M and simulates between time 0 to time 10 in 100 substeps, if I write it like that. And there are a bunch of options for that simulator that I can change. If I were to print S, and we can do that here, we can go into our antimony here, and I can just print S. It will give me the object, which is the output of that simulation. And I'll see it's actually a rather complex object. It's got the column labels at the top, and then it has the time in the first column, and then the values of the 2 variables T and I in the second and third columns. And then there's a simple method plot, which is defined in Tellurium, which takes that simulation output object and puts it up in a plot of the form that we have here. There are lots of options that you can do to change the colors of the lines, to change the width of the lines, to say whether it's points or lines and so on. We're going to use some of those. We're also going to wind up using matplotlib plots a lot instead of the Tellurium built-ins. So that's what well, I'm behind by a minute or two, but that's the main things that I wanted to do. If we had a little bit more time, what I'd ask you to do, and maybe you can do that in just 10 seconds, is you can go into your simulation here and get the size of the window to work. Go to the simulation here, and you can change one of the parameters. For example, you could make the amount of virus smaller by a factor of 10. And you can run it, and you can see what happens. I'm not sure why this is a zoom interaction issue. If I make the amount of virus less, I should have, I'm sorry, I don't know why the screen is not behaving that way. If I make the amount of virus less, the rate of infection is less and it stretches things out. And so I would encourage you now to take this little example when you get home and try changing the parameters and seeing what happens.